Okay, so we are starting with chapter 3, which is all about the cell. And we're going to talk about, during this chapter, cell structure, definitely function, because that's the physiology part of this, movement across membranes, cell division, and stuff in this here. And I'll divide it down into sections like I've done before. All right, jumping right in then. The cell is important to living structures, to our human body and everything, because it is the smallest, the basic unit of structure and function in living organisms, including in your own body. So anything smaller than a cell, so you can look in here in this picture and you can see little organelles, you can see a nucleus. Those are all necessary for cell function. So they each have a job within that cell. But by itself, the cell, one cellular unit, is the smallest single unit of structure and function for living organisms. Because remember that we have the smallest units of structure, like for an atom, and you have different things, matter, and that kind of stuff. This is, the key here is living. So if I get smaller than this, I don't have a living organism. All right. And there's some basic things that you have to have around it, or as part of every cell. So we're going to go through the basics, and then we're going to go through specific structures within the cell. Keep in mind, as we're looking at this and as we're going through these cells and everything, when you look at a picture like this one, this is what we call a standardized cell. So it's an animal cell that has just like basically everything in it, so that we can talk about each of those structures. But your cells are specialized. So you have neurons that transmit impulses. You have muscle cells, you have epithelial cells, and that kind of stuff. So they're going to have more or less of these structures, or they're going to have modified structures within the cell. But we're going to start with the overall picture and then work with that, okay? We have a ton of cells. You start off as one single cell, the egg, that got fertilized by another single cell, a sperm, and you became a little zygote. And the zygote starts to split, and you get 2 cells, and then you get 4 cells, and then we get more and more cells, and you become this little ball of cells. And then those cells at some point start to differentiate. They become specialized into tissues. And so that's when you start developing, and it's very, very early on, by the way. It's very quick. All of this is happening within the first few days after fertilization. But then you start to specialize, and you're going to start building muscle tissue and bone tissue and all that kind of stuff. By the time we're done, we're talking about trillions and trillions of cells. You have about 50 to 100 trillion cells. So million, billion, trillion. This is a lot of cells. And throughout your lifespan, you're constantly going to be replacing these cells and getting new cells. So there's a lot going on. Remember, every time that you make one new cell, you have to copy everything that's inside that cell and make it perfectly also, to so it'll function correctly. So I kind of said something about differentiation in the last slide. So let's define what that means. Differentiation is basically just the process of becoming specialized. When a cell differentiates, it goes from being a generalized cell to a cell that has a specific function. So a nerve cell, it does one thing. It transmits impulses, or it stores memories or whatever, but it's for the nervous system only. Muscle cells, they contract, okay? They're not going to produce all these enzymes and stuff that you would find in the digestive system. That's a whole other set of specialized cells. But what we get then is we get cells that have different shapes, different structures, different sizes because of their function. Remember, structure determines function, so the cells themselves don't look like this perfectly little round cell that you see in all the diagrams that says animal cell. They really have specialized structures to fit their function. This one you can see in this picture, this is a nerve cell, and look at it. I mean, it looks totally different. Right here, crack that voice just a second. Right here, you can see what looks like a generalized cell. So there's the cell body, but then you have all these other little projections that come off of that. This is so that this cell, which is a nerve cell, can receive information from multiple sources, and then it can conduct that information out and again send that information to multiple cells too. So it's highly specialized for receiving information and sending information to other parts of the body, from one part to another part. These are red blood cells. Red blood cells are specialized cells that carry oxygen around the body. They start out as nice round cells, and then you can see this little sort of caved in appearance right in here. They eject their nucleus as they mature so that they can become specialized. This little space here allows them to bind more oxygen. So they're specialized to do their function, and then they float around in the body for a limited number of days. Their job is, like I said, carry oxygen. Past that, since they don't have a nucleus, they really don't have the ability to create more of themselves. Like many other cells will go through what we call mitosis, cell division, and create more cells. Red blood cells have this determined lifespan. They're here, they do their job, and then they're destroyed. We have to make a new continuous supply in the bone marrow. These guys cannot make copies of themselves. This is muscle cells, and you can see that these cells are really elongated strips, kind of looking to that because they need to contract and kind of pull in for that. Things that we'll look at when we start looking at tissues is kind of little features that stand out. This one, you can see this line right here. This is called an intercalated disc, so that is a cardiac cell. And then you can see these lighter looking lines here. Those are called striations. That tells you have muscle. There's a nucleus, that kind of stuff that you can see in there. But again, specialized for contraction, so they're very elongated to do that job, to be able to get shorter and get longer and move things. These are what we call epithelial cells. These are actually from the cheek. So the inside of your cheek, it's a lining, a cell that is a lining, so it's very thin. You can see that they're sort of rounded, they're thin. This one's kind of odd shaped, but because they're so thin, they fold over on themselves. You can see the nucleus in here. They've been stained so that you can see them. Again, these are lining, so they're very thin and round. You lose them a lot, you replace them a lot. So then going back to our typical or our composite cell, like I said, when we look at it, we're going to just put everything together into one cell and say this is our cell, and this is what we have. Every single cell, at a bare minimum, has to have a cell membrane. You also might see plasma membrane as the name, but that's the surrounding of the outside of the cell that divides the cell from interior and exterior of it. In humans and in other what we call eukaryotes, things with nuclei, you have a nucleus in here in the cell that contains the genetic information, and then you have cytoplasm. And cytoplasm is this fluid material in here that contains all of those little organelles, or those little specialized organs inside that the cell that do specific jobs for the cell. Let's start with the cell membrane, and then we'll work to the more complex organelles and stuff. The cell membrane, again, its job is to be the boundary between what is in and what is out, and what can come in and go out too. So it's the determination. What do I want in the cell? What do I need in the cell? And what do I need to get rid of? Like, did I make a product, like an enzyme for digestion? Then I will need to get rid of that product so that it can be used elsewhere. Or do I have waste products? I'll need to get rid of that. So things going in and out of the cell are regulated by that cell or plasma membrane. We call it selectively permeable because permeable itself means just moving in and out. But the cell determines what and at what quantities things can move in and things can move out. So it's selectively permeable. It says, I need a certain amount of this that needs to come in, or I need to get rid of this, but not everything. So everything doesn't move freely in and out of the cell. It's like the gatekeeper. It says, you can come in, not you, or this much of you guys. It's the gatekeeper for what goes in and out. Notice on here, it is this like double layer. You see this layer here and layer here with these tails. We talked about phospholipids earlier. We're going to get more into that in a minute. You can see that there's things embedded. They've got them colored purple, so proteins and some carbohydrates. So it's a pretty complex little structure. So let's begin with the basic structure, and the primary structure is what we call those phospholipids, and I talked about that in the last chapter. Remember, the phospholipids are the phosphate group with the two lipid tails, so they're a special type of lipid. And I brought in the terms hydrophobic and hydrophilic and that kind of stuff. They are permeable to lipids, so other lipids, so anything else that's fat can move pretty freely through between and past these guys. The phosphate groups, if you remember from the last chapter, are hydrophilic, meaning that they like water. So they're happy to be around water. They are soluble in water. So this part here, that's the phosphate head, that can be around water. These two tails down here are those hydrocarbon tails. They do not like to be around water. These are the parts that's soluble to lipid soluble substances. Okay, so you have this conflict going on in here. This part likes to be around water. This part does not like to be around water. So that actually creates a very nice barrier between inside and outside. There's the lipid tails. I talked about it. They are hydrophobic. They are made up of hydrogens and carbons, so they're hydrocarbon tail. And they, like I said, don't want to be around water, so they're going to do their best to get away from water. And what they do is they face the interior of a cell. What we're going to have is, since this part, the phosphate head here likes to be around water and the tails don't, you end up with this double layer where they face towards each other. So the tails come in contact with each other and the phosphates face away from each other. And then you just build this layer, and it's in 3D, I'm drawing in 2D, but it's faced like this around. So I had this interior section that is water-free, and then this exterior section that can be the outside of the cell or the inside of the cell that can be around water. Because remember, you're 2/3, I should say, 2/3 water. So you've got to have the ability to have your cells in contact with water. There's water inside the cells, there's water outside the cells. So you have to have something that allows them to be around water. That's the phosphates. And then you want a barrier that prevents stuff from moving through. That would be the lipid tails. So let's go here. So again, phosphate head. It is hydrophilic, starting to get to know these terms. Another term that goes with that is polar, because it has a charge. So you have this polar. Anything that is polar is hydrophilic, by the way, likes water. Then you have these tails here that are really just the long chains of hydrogen and carbon. They are hydrophobic. So hydrocarbons are always going to be hydrophobic, and they are also always going to be nonpolar. So again, start associating all these words together. All right. And then when they form together, so there's one at the top. When they form these long sheets of, here's the heads, here's the tails, one sheet and then another sheet. So what we call this is a bilayer. It's a phospholipid bilayer because you have one layer here, right, and one layer here. I have and this could be the inside of the cell, this could be the outside of the cell, or vice versa, but this whole thing then is one cell membrane made up of two separate layers of these grouped together phospholipids. That is the basic structure. So that's what we start with. So the phospholipid bilayer is what we start with. And then the cells are going to embed components. So what we end up with is something that we call the fluid mosaic model. Fluid means something flows. So it's not a solid structure like a tabletop or something. It's actually a structure that the cell membrane is able to move components around, and it can move quite a bit. So it's not this hard, solid structure. It's really this flexible structure. It has this fluidity to it, like a liquid does. Mosaic, because it's not just one thing. A mosaic picture, like in Greek and Roman times, was where they took little pieces of glass or tile, and then they would put them all together, and when you step back, it's a big picture, right? But when you're up close, it's just all these different colored tiles. So the cell membrane kind of looks like this mosaic of different embedded structures inside that cell membrane. The basic structure, remember, are those phospholipids, bilayer, on that end. And then embedded, I'm going to have proteins, I'm going to have cholesterol in between. So mostly phospholipids with a bunch of other stuff, sort of part of that layer, or those two layers. So cholesterol is one of those things. Cholesterol is necessary to your cell membrane because it gives it a little bit of rigidity. Remember, we've got this layer that's fluid. We don't want it to be too fluid. You don't want to just like have your cells just flow away. We've got to give them a little bit of structure, a little bit of stability, and that's what cholesterol does. Too much cholesterol is going to make it too hard and too much stability so that things can't move through the membrane. So it's this playing this game of I need enough cholesterol, but I don't need too much cholesterol. And we as animals, all animals make cholesterol. So you make your own cholesterol that you can embed in the membranes. And then when you eat another animal, so meat products and stuff, you bring in their cholesterol. And that's where we run into that problem of high cholesterol and stuff. So it's not, it's not bad to eat meat and get their cholesterol. You just got to watch how much you get. And then if you're actually genetically prone to making too much of your own, it becomes this whole issue of, building on itself. You just kind of have to watch that. But it is necessary in the right amount. The other thing that is embedded in here, or one of the other things, is a group we call proteins. Remember, those are one of our organic molecules, or complex molecules. And they can be all kinds of things. You can have one like I drew up here that goes all the way through, and so it acts like a channel that allows stuff to move through that. So they can be pores or channels, or even these little, what we call carrier proteins, so they can physically carry things through the membrane. They can be on one side and act like little receptors, so any like hormone or something that's coming to and are circulating around the cells can bind to it, so it can initiate an action on the inside of the cell, tell the cell to do something. In other words, like make some sort of protein or make something. They can be enzymes embedded in that membrane. We're going to come back and talk about CAMs on a separate page in here, so I will specifically talk about CAMs, but basically CAM just stands for cellular adhesion molecule, and they have specific jobs. And they can also be, very importantly, these little markers that we call self markers. You have an immune system that's job is, these cells run around your body, and their job is to identify cells that shouldn't be there. So things that could be dangerous to you, bacteria, viruses, fungi, whatever, little cells of parasites, whatever it is, and then destroy those things. So those cells need to know the difference between you, your own cells, and these foreign cells. So a lot of these little proteins that are embedded in your cells are little markers that just say, hey, this is me. You recognize me, leave me alone, don't kill me, go find somebody else. So they're identifying self from non-self for your own safety. When they forget this, when they kind of lose this ability to recognize self, so the immune system stops recognizing these self markers, that's what we call an autoimmune disease. So your own immune system starts to attack your own cells, and we want to avoid that. Okay, so here is what we call the fluid mosaic model, right? So the yellow would be the phosphates. These little green, that's the lipid tails. Notice that there's two layers, and they face opposite directions. So the lipid tails are on the inside of the cell membrane, and the phosphate groups are on the outside. That means I can have water here, and I can have water here, so inside the cell and outside the cell, but there's no water in that cell membrane itself. So this little area right in here is a water-free zone. So there's no water there because the lipids don't like it. But again, that creates this barrier. So only some things can really travel through those phospholipids. Anything that could be soluble in those lipids, in other words, mix in with those lipids, so other lipids, right, are the only things that can really move through there, or very small things. Otherwise, they're going to need help. And they get help from these things like this right here. This protein is so it's called a transmembrane. It's also called an integral. That's what it's usually referred to, or I hear it referred to. An integral protein. So it's those things like those pores and channels and stuff that allow substances to move through that cell membrane, bypassing those little lipid tails and not having to come in contact with those lipid tails. So remember, the middle is hydrophobic, where you have the lipids. The phosphate heads in yellow are hydrophilic on both sides, okay? And then we have the protein that goes all the way through. That's an integral protein. And then we have these proteins called peripheral. So this right here would be like a peripheral protein. Your periphery is like on the outside. So your peripheral vision is not what you see straight ahead. It's what you kind of pick up on the sides when somebody walks up behind you. This is what that means. It's on one side of the membrane or on the other, but it doesn't travel all the way through. So these things are the things that could be those self markers. They could be the receptors. They have a lot of different functions. All right. So what happens? So here's like a channel, like this guy right here. Say this is one of our channels, and it's supposed to allow something to go through, right? So I need a specific ion to go through, maybe like calcium to run through it. What happens if this is not built correctly? Are you going to get the same result that you should? If I change the structure, I change the function, and that's a problem. And all proteins, by the way, are coded for by your DNA. So it would go back to your DNA being in it, coding for an incorrect protein. So what we call that is something that's faulty. It's not working correctly. So we made it an incorrect ion channel. And here's some of the diseases that can go along with that. Some individuals feel no pain, okay, and some individuals feel too much pain. What happens is pain is picked up on in your nervous system. You have specific receptors, like say you cut your finger, right? You have specific receptors that are just for pain, and then they send this impulse all the way up the nervous system to your brain, and your brain says, that really hurts, stop doing what you're supposed to be doing. That's how it works to protect you, right? Well, what happens if that signal never gets to the brain? You are doing damage, you are hurting yourself, but your brain never picks up on that signal because it never got there. So the problem is that the sodium channels that are responsible for sending that message, sending that impulse along the neuron, they have a mutation. So they're not built correctly. Change the structure, change the function. So if I have a channel that is blocked and does not allow sodium to go through, so normally, here's my little channel, I want sodium to run from one side of my cell to the other side of my cell in order to help transmit that signal along the neuron, right? Well, if that channel is blocked, then I can't, that sodium's never going to pass through. There's no pain message being sent to your brain, so your brain never gets the message. You never feel pain. And you think, well, that would sound great. But the problem is pain is healthy. Pain is one of those things that says stop doing what you're doing because you're creating too much damage. So if I hurt my fingers, I put it on something hot or I cut it, that my reaction is to pull away from that, and it limits the amount of damage. I have some damage, but I don't have a whole bunch of damage. I limit the amount of damage so that I can stop doing what I'm doing. Conversely, if those channels are open too long, you're sending too much message. It keeps sending the message, and that person's going to feel excessive pain because they don't just get the message to the brain that, hey, that hurt, stop doing it. They're getting the message again and again and again and again and again. So they feel exaggerated amount of pain from that. Another one is called long QT syndrome, and I've got a picture somewhere. Okay. This is recognizable as like a heartbeat, right? So if somebody takes your, they're mapping out your heart and they take a little EKG on you, you have these peaks, which means it goes up, and you have these valleys, which go down. And everybody should have a standardized sort of look to this. And we call these peaks, sorry, it keeps coming up. These peaks, we label them with letters. So P, Q is that valley there. R is another peak. S is another valley, and then T. Notice they go along and they go up and down and stuff. And they should be not only have them at certain lengths from each other. So P to R should be a certain length. They should also be at certain heights, right, or depths. So that indicates that there's something wrong or there's something right. With long QT syndrome, it's the problem between the Q and the T and the distance between that. When you measure that, what's happening is this time period between those two is actually extended in somebody with long QT. So instead of going from right here to right here, it's actually the T that it peaks later on. Okay, and that's a problem. Okay, that's why we measure those. We can see that that's a problem. And this one has to do with an ion called potassium. Potassium is also important in nerve impulses, but it's also important in muscle contraction. So what you see then is problems with muscle contraction, heart problems, irregular rhythm. You can also see damage to hearing. So they have some impaired hearing due to possibly excessive potassium that causes stress on those receptors and then damages those receptors. Cystic fibrosis, CF, is a genetic disorder that is the most the most lethal for Caucasian Americans. Okay, so each group, based off our ancestral history, where we came from, generally genetic mutation is a random act, and it gets into a group. So back when we lived in small towns, villages, whatever, if a mutation got into that small town, you just really had those people to marry and grow up with and live with, and then your kids married in that same group. So you tend to inbreed those sorts of mutations on a larger scale, not on like a familial scale, but still within that village, if it's small, or that small town or whatever, and it stays within that group. So you see like Caucasian Americans because of the ancestry from Northern Europe or from Europe. You see African Americans because of the ancestry from areas in Africa. You see Asian Americans, and each of these groups, even Eastern and Western Europe in there, you see different genetic disorders that are common to those groups. They're not only in those groups, but they're more common in certain groups. Cystic fibrosis originated in Northern Europe. So when we had people come over from Northern Europe and colonize the U.S., they brought with them this cystic fibrosis gene. And cystic fibrosis is a problem with chlorine in the chlorine ion that is necessary to help with breathing. What happens in this is you have a protein channel in your cells that allows chlorine to move through, and this aids in breathing. It allows you to have the right amount of mucus in your lungs and to pass the gases, the oxygen and carbon dioxide, through the tissue so that you can transfer those things. Well, the problem here is too much chlorine, and the problem is those channels aren't working correctly. So what they get is this really large amount of mucus. We all want a little bit of moisture. You can't have gas transfer through tissues without moistened tissue. So we have a little bit of moisture in our lungs, but due to this imperfection, this gene, this genetic disorder, there's too much mucus being produced. So too much liquid, very, very thick. So it's the wrong consistency, and it just gathers in the lungs, and it makes it impossible to breathe. It's like drowning in your own body fluids. And so to get rid of it, you have to pound on the chest with these kids, pound on the back, and basically loosen it up so they can cough it up. Another thing that you can see is the salty taste, and that comes from sodium chloride, right? It comes from the chlorine, the excess chlorine, so you get this kind of salty taste going on in there. But over a period of time, it does a lot of damage, and it's really visible in lungs because breathing, but it's also visible to a doctor and stuff in the pancreas as the pancreas starts to shut down. And it's, like I said, it's lethal. So, without medical help or medical treatment, these kids usually can die pretty young in the United States, 20s, 30s. Sometimes they have a very mild progression and they can live a full life, but they still have to have medical treatment the whole time. CAMs. I told you I would talk about CAMs. Here's the CAMs. Cellular adhesion molecules. These guys are little molecules that are located on cells. So you see the cell right here, this white blood cell. It's not the white blood cell itself. It's the structures that are part of the cell. So it's on that cell membrane. And their job is to help with cell movement. White blood cells don't stay in one place. They roam your body looking for pathogens, looking for things that might harm you, right? And they try and destroy that. They don't just kind of flow through the bloodstream just freely, kind of like red blood cells do. These guys actually move. along the cells of the bloodstream. So this picture here, this is a capillary, which is your smallest blood vessel, and they're going to move along here and be guided by these things. So it's like an attachment mechanism. They attach and release along the way. Okay, so there's two types, integrin and selectin. WBC means white blood cell. So the white blood cell, when there's damage here, it gets this sort of instructions from the damage. You've got all kinds of chemicals released like heparin and stuff. And so the white blood cells will not only rush to that area, the integrin causes the white blood cells to change shape so they can sort of squeeze out between the cells. They get really, really thin and they can just squeeze out and then get back in their regular shape on the outside and go find whatever may have entered through that injury. So some bacteria entered with this splinter right here. The white blood cells aren't stuck inside the bloodstream. They can exit into that surrounding tissue and mop up, clean up whatever could possibly be dangerous to you. Selectin is what I was pointing out right here. This is those things that anchor or hold onto. So they like clasp and let go, clasp and let go, so that the cells don't just flow through the bloodstream. They move on those cells themselves. And then there's other secondary types of uses for this. They're also important in embryonic tissue, growth of that, and then growth of nerve cells too. All right, from cell membrane to cytoplasm. The cytoplasm is anything from the nucleus, not including inside the nucleus, but from the outside of the nucleus to that cell membrane. So it's just all of this territory in between. And it's made up of really two things, and we kind of use them interchangeably sometimes. Cytosol is really just the liquid portion of the cytoplasm. A lot of people just say cytoplasm to refer to the cytosol, but really cytoplasm is the cytosol plus organelles, and organelles are the little organs. They have specific jobs within the cell. So you see this yellow one produces energy. This purple one right here is going to package protein. So they all have their own little specialized job. But the cytoplasm itself spans from the nucleus to that cell membrane. So once something enters the cell, it is in the territory of the cytoplasm. As far as some of the major organelles that we're going to talk about, the first one is easily called the ER, but that stands for endoplasmic reticulum. When you look for it, see the nucleus right here in blue, endoplasmic reticulum usually comes right up next to the nucleus and then starts spreading out from that. So it's like tight up in nucleus and then it gets looser as it spreads out from that. But basically, they're just like these flattened little sacs that that transport substances from one place to the next. There's two types of ER. There's the rough ER, and it's called rough because it looks rough, and that contains ribosomes, and ribosomes produce protein. So their job is to produce protein. So if a ribosome, these little dots on here, produce protein, they secrete that protein inside the rough ER. That means the rough ER then has something to do with protein. So really what it does is it transports and modifies those proteins. The smooth ER is called smooth because it doesn't have the ribosome, so it looks smooth. It also looks more tubular than the rough ER. Its job is to deal with fats. So it makes fats and stuff for the cell that would be used for different uses in the cell. It also has a secondary job of breaking down, not secondary, but another job of breaking down drugs. So you find a lot of the smooth ER in places like the liver, where you metabolize your food and things that you brought in, and it sort of cleans things up and keeps you from poisoning your cells. Like I said, the ribosomes themselves, these little dots on the ER. So the whole thing here is the ER, the ribosomes, the little dots. Their job is to make proteins. So they make proteins. That's called protein synthesis. They're found associated with the rough ER. They're also found by themselves out in the cytoplasm. So they may not actually be attached to anything. We call that free floating. The Golgi apparatus, or sometimes called Golgi bodies, are here, these little purple sacs. They look a lot like the ER. By the way, these colors are not real. We falsely color them so that you can tell the difference between them. But in this diagram, they're purple. So once the ribosomes have made protein, they've deposited them into the ER. The ER has modified them. They will then ship out those proteins to the Golgi apparatus that further modify and package the proteins. They're kind of like UPS. They take those proteins, they package them, and they tag them so that if those things are going to be exported out of the cell, those proteins know where they're supposed to go. So other cells can recognize them and pick them up. They're transported within the cell in these little things called vesicles. And a vesicle is just a little portion of one of these organelles that broke off, and it contains something. So like the box, like a box for UPS, right? So it's what contains something on the inside, and it can contain all kinds of stuff. It can bring in stuff from the outside vesicle, and part of the cell membrane becomes the vesicle, or it can be one of these organelles that becomes a vesicle. Not the whole thing, just a part of it pinches off. So there's the vesicles. Like I said, here's one coming off the Golgi apparatus. You can have them coming off the ER, and they basically just transport stuff to the cell membrane. You can see where this one is pinched in right here. That vesicle became part of the cell membrane, and then stuff just was released out. Mitochondria's job is to make energy for the cell, and that's in the form of ATP. So it converts food energy, it uses oxygen, and it makes our usable form of energy for the cell, ATP. There are lots of folds, there's lots of partitions. It's a double membrane structure. So you can see in orange, there's one membrane on the outside, and then there's this internal membrane that is really folded. Here's another theme in biology, surface area. This has a large amount, by folding this inner membrane in, it has a really large amount of surface area. That means it has a lot of ability to do something. And in this case, a lot of ability to make ATP. Your brain is folded, a lot of ability to comprehend, to analyze, and to store information. Your intestines are folded, a lot of surface area in order to absorb food particles and break down food. So by folding things, we can increase surface area and put them in a small place. The mitochondria is like that also. The folds, this little portion that bends in, these are called cristae. Okay, so that's what that term is. Lysosome is a little sac, another little sac, like a vesicle, little sac, but inside of it, it has these special enzymes that help break down substances. So you can see here, it's made up, all of these things, by the way, are made up of a double membrane. So just like the cell membrane is this phospholipid bilayer, all of these organelles also are phospholipid bilayers too. So it's all part of the same thing. But this one contains enzymes. It has a low pH, so like an acidic pH. Basically, its job is to break down things that shouldn't be there. So destroy things that are either outlive their usefulness. So like an old mitochondria that's no longer producing enough ATP, get rid of it, recycle the parts and make a new one, right? Or bacteria or something that might have entered into the cell, they can surround it basically and digest it. They can break down food particles so that the food can be used. They're sometimes referred to suicide sacs since they do break down their own parts of the cell. Also like a worn out mitochondria or something. A specific thing that's very similar to a lysosome but has a specific job is a peroxisome. So you can look at the picture and it looks just like a lysosome. It's a lysosome with a specialized job. So they also contain enzymes to break stuff down. You see them more often in places like the liver and kidneys because they have these multitude of jobs. One of that is to break down peroxide. There's its name, peroxisome. It breaks down hydrogen peroxide, which unfortunately your cells make, but in large amounts it will kill the cells. So these enzymes break it down into water and oxygen. Water is not dangerous to your cell. Oxygen is not dangerous to your cell. So, so you have this ability to break down something that your cells normally make that could kill them. It also makes bile acids, which are help to break down lipids. So that's part of your digestive process. Bile breaks down fats for your cells. So the bile acids help to break that down. It gets rid of biochemicals that could be dangerous to you. So that's why you see it a lot in the liver. So as soon as you digest something from your small intestine, it's going to move into your bloodstream and go straight to your liver. And then these peroxisomes, the cells in the liver contain a lot of these peroxisomes that will break down biochemicals and alcohol that could potentially kill your cells. Another organelle is called a centrosome, and these are specifically found in animal cells. So this is something you would find in your own cells. Their job is to help out with mitosis. So they take the chromosomes, they separate out the chromosomes. The rest of the chapter, later on in the chapter, we're going to spend a lot of time on the actual process of this. But they help separate chromosomes during mitosis so that each new cell that is formed will have, the right number of chromosomes inside of it, and you're identical on those two new cells. They're also a part of moving structures that allow cells to move, like cilia. Cilia are little hair-like things that sweep stuff. So you have them like in your trachea, and they can sweep out the dust out of your trachea and your bronchial tube so that you don't get a bunch of dust down in your lungs. They line like oviducts and stuff so that you can move the egg along through those tubes. They're also part of the flagella. The sperm has that little tail-like structure that swims, and these tubules here are made up of these. The tubules make up those structures. Cilia, like I said, is hair-like. And you can see the electron micrograph right here, and it lines one side of the cell. So you would have like another cell here and another cell here, and they just line here. So any dust that comes through, it can sweep it back out, right? Very hair-like, small. They kind of work in unison. They just move substances along the surface. The flagellum is like the tail. So here's the sperm. The yellow is the head. The tail is right here, and that just propels that sperm along. You can see all those tubes that we were talking about earlier right there that allow for that movement, that structure of that flagellum. Flagellum is singular. Flagella with an A is the plural form of that, just so you know. Microfilaments and microtubules are they're found throughout the cell. Their job is to support the cell membrane, to give structure to the cell, shape to the cell, okay? And organelles will actually attach to that and get sort of in between and kind of anchor themselves in there and stay in one place. Or some of them, like mitochondria, will actually move from one area to another area, but they anchor within those or between those microfilaments, microtubules. Some organelles will attach to them and they actually move along there. Vesicles that come off of your Golgi apparatus don't really just float through the cytosol to the cell membrane. They have like a railroad track. They move along these microtubules till they get to the cell membrane, and then they can release their substances to the outside of the cell. And then extra things. Inclusions are just like extra things. So little things that may be there at one point may not be there. Temporary things like color, pigments or colors, nutrients come and go in there. So these little blue in here, they've got them stained, are like little inclusions in the cell. And it's just, temporary types of substances. All right, let's talk about some diseases of organelles. There's a lot out there, but remember, structure determines function. So if I don't build one of these organelles correctly, it won't perform whatever function it's supposed to. One of them that's really they're all tragic, but one of them that is really tragic is this thing called MELAS. And MELAS is short, even though it's like 5 letters long, short for mitochondrial. So we're talking about ATP production, energy production right there. Mitochondrial, encephalomyopathy, lactic acidosis, there's the LA, and stroke-like symptoms. So all of that tells you that this does not sound good, all right? First of all, we're dealing with not making ATP. So the cell doesn't make energy And without energy, the cell's going to die. So you're going to see all of these other things that go along with that because the cell's just not getting enough energy. And you end up with symptoms like migraines and strokes. Diabetes is not caused solely by this. But this can lead to diabetes. Muscle weakness because there's no energy. It is in the mitochondria. You inherit pretty much all of your mitochondria from mom. So this genetic disorder is actually inherited from your mom because your mom gave you all of your mitochondria in her egg cell. The dad's sperm does not usually contribute the mitochondria to the child. Tay-Sachs is another really tragic one. Tay-Sachs is seen very common in Ashkenazi Jewish populations and Cajun French and Canadian French. And again, that's that inheritance thing that I told you about, going back in our history to small villages and small towns and stuff. If you're Jewish traditionally, and we're getting away from that, but Jewish traditionally, especially really strict Jewish types of groups, you stay within your group. You marry another Jewish individual, sometimes within the same area, the same town. Cajun French and Canadian French are the same way. They grow up in these small towns. They're born in that town. They grow up in that town. They marry in that town. Their kids stay in that town, and it's perpetuated in there. So if a genetic disorder randomly shows up within that population, it just gets bred back into it because you have a small gene pool to choose from for potential mates. And Tay-Sachs is one of those. So it showed up as a mutation in these Jewish groups, and it also showed up as a mutation within French groups that ended up in Louisiana and in Canada. All right. It is inherited. In this case, both parents contribute. It doesn't come from one, like with Mealis, it comes from mom, right? This one, you have to have both parents contribute a gene to this, and it deals with the lysosomes. So the lysosomes are not built correctly. And their job, remember, is the garbage men of the cells. They get rid of waste products. Well, these guys can't get rid of the lipids. So it takes a while to see it. So your cells start to accumulate lipids. In a normal person, you would accumulate lipids and your lysosomes would destroy it and get rid of it, and we're all good and everything stays the same. Well, these kids, babies, start to accumulate a little lipids. Their lysosomes can't get rid of it, so they accumulate a little more and a little more and a little more and a little more. So tragically, what you see is this baby that is going through all the milestones that a baby would. It's sitting up, it's rolling over, it's eating food, it's starting to walk, and then there's this regression. It goes backwards. It starts losing the ability to do all that. And that's because it takes a while for enough lipids to build up to start showing symptoms and damage to cells, specifically damage to cells in the brain. And the brain is going to control all those actions. Every action that you do, as far as walking, moving, talking, swallowing, digesting and all that, comes from information from the brain. So once those lipids build up enough, they cause damage to the brain, and then you see this regression. There is nothing we can do about it. These kids die. They die by the age of 5. We can't stop the progression. We can't make it, basically you can make it a little bit better towards their death, but we can't cure it. There's nothing we can do about it. One of the symptoms that they do show is this red spots in the eyes. And so that's kind of one of those earlier signs in that. So here's the thing is you inherit 2 normal people, right? They have a gene for it, and they're not showing any symptoms because this is what we call a recessive gene. So it's hidden in them. They get married and they have babies, right? And what I'm drawing is a Punnett square here. For those of you, it's been a while since high school. So they have a chance. These are the chances. Each part of this square here is the chances that they would pass along this gene. So the capital A is normal. So they have a 25% chance that each parent would pass along a normal gene and they would have a normal child. They could also have a chance of passing on one or the other gene. So this parent passed off the abnormal gene. This parent passed off the normal gene, and this one passed off the abnormal gene here, and then here it's this one normal and that one abnormal. So a 50% chance that they would have a kid that was considered a carrier like them, normal in appearance, but have the gene that they could pass off. But every time they have a child, they have a 25% chance of having a child that has this disorder and would die from this disorder. So if it's carried within that group, then there's a chance that you would come across another person. So for like the Jewish and the French populations in those areas that I discussed, 1 in 30 people are carriers. They have this capital A, little a genetic makeup. In our normal population, it's like 1 in 300. So the chances that you would have it is 1 in 300. And the chance that you would come across another person that has it would be 1 in 300. So the math there is not very likely that you would have 2 people come across each other. But when you move it to 1 in 30, 1 in 30 chance, when you grow up in these small little villages and you're culturally from that group, 1 in 30 chance, and then 1 in 30 chance that your spouse would have it. So it becomes a lot more likely that it gets passed along within that group. Horrible, horrifying. Watch your child die, basically, is what you're doing. ALD is one that you guys are probably familiar with as far as the name, adrenoleukodystrophy. This one is a problem with the peroxisomes. So the peroxisomes, who are also a cleanup crew, right? They break down lipids and stuff. They also can't do this job. And so they can't break down the lipids, but the myelin that's found on nerve cells, myelin is on nerve cells and it speeds up the rate of an impulse. Well, this particular disorder starts stripping away the myelin so you can't transmit nerve impulses, okay? So again, this child starts out with normal development, starts to grow up, but about the age of 7, 8, somewhere in there, they start showing symptoms because of the stripping of the myelin, and nerve impulses aren't going to where they need to go. And then it's a regression until death too, usually 14 or 15 years old in there. And it's a horrifying way to watch your child just become bedridden and die from this. In the 1980s, early 1990s, I think it was 1980s, there was a family whose child, grew up, they found out he had it, and they were told that there was nothing they could do. This child was going to die, and they just had to watch him die. His name was Lorenzo. Showed all these symptoms here: weakness, lethargy, darkening of the skin, behavior problems. He became bedridden and everything. But they absolutely refused to believe that there was nothing that could be done about it. And the dad actually did all this research, and he brought together, the problem with the 80s was no internet, right? So you had research groups in one part of the world working on one piece of the puzzle and another part of the world working on another piece of the puzzle, and they weren't really able to communicate and put all this together. Well, he took all of these research pieces and he put it together, and he actually discovered, this guy wasn't even a doctor or a research scientist, discovered that there was a certain type of oil that you could provide in the diet, and they called it Lorenzo's oil for his son, provide in the diet, and it not only started to reverse some of the symptoms, it halted the symptoms in kids. So if you started it early enough, these kids would never go through that successive weakening and death. They could actually live a normal life. By the time it was discovered, it was too late for Lorenzo. But Lorenzo has, as far as it stopped the progression, and he's had some repair, but he can't go back to normal. So it was too much damage was already done. But they have managed to save all of these kids from this because, sorry, I get worked up about it. It's so sad. They have managed to save all these kids from that time on by changing their diet, because he refused to believe what all of the doctors and all the scientists were telling him. Great movie, but bring a lot of tissues if you watch it. All right, cell nucleus. Cell nucleus is the control center. Its job is to tell the rest of the cell, what are you supposed to do? And it does that through containing the genetic material, the chromosomes, that have the instructions for building things. It is made up of an envelope. So we have this nuclear envelope, which is a phospholipid bilayer. It is a double envelope. You can see how it loops around here. So there's 2 layers. So not only is it one phospholipid bilayer, it loops around and becomes a double phospholipid bilayer there. It does have pores that allow things in and out of the cell, right? Or not out of the cell, sorry, out of the nucleus. You have structural things that need to enter the nucleus. You have information that needs to get out to make proteins. So you have different things that need to pass through. Just like a cell, it has its own cytoplasm type of thing, but since it's not cytoplasm, we call it nucleoplasm. Okay. Inside, you can see this dense region right here. It's just really a dense region of a lot of RNA. That's called the nucleolus, and its job is to make the ribosomes. So the nucleolus makes the ribosomes. The ribosomes then have to leave through the nuclear pores. That's why they're one of the reasons they're there, and go out to the cytoplasm where they can then do their job of making proteins. But they're first made in that nucleolus inside the nucleus. And then also in there is the genetic material. And we have these names called chromatin, chromosomes. When it looks like this, see this looks like a big spaghetti bowl. It's unwound, it's loose, it's actually doing its job, and that's when we see it. Most of the time it's like that. It's referred to as chromatin. It's these names, chromosomes and chromatin, are just a difference in structure. They're not a different or shape. They're not a difference in components. So chromatin is DNA, and it's got some protein components. Just before the cell divides, it actually shortens and thickens and becomes these little packages, and those packages are what we refer to chromosomes. You guys are more probably familiar with seeing these chromosomes because this is what we show all the time when we study DNA and everything. But really, in reality, this chromatin is how it's normally in the cell, in the nucleus. It only becomes this little X-looking structure during cell division, during mitosis. But you can see in here, it does have a protein component, little histones. There's DNA, the double helix that you're familiar with. So chromatin by itself is not just DNA. It's DNA that's wrapped around these little histones and organized around these little histones. And that's going to be it for this chapter, or for this portion of this chapter. I'll pick it up in part 2.